So, in this experiment, we are going to check the what is the effect of different chemicals or the drugs on red blood sugar. Now, before we do the experiment, I need to show you what is the principle involved in that, what is the chemicals it is that. Say we are doing the experiment on the uh, virtual lab software. It is not the like that of the original handling of the animal. So first I'll discuss the principle and all that, then I'll discuss about the procedure, how we can do the experiment and what is the difference if you are going for the this particular experiment on the animal. That means if you are doing some new research and if you are using the animal, how we have to do this experiment. So the experiment on the blood sugar, so click it, so go to the virtual lab. Now say what is that here. First we need to understand that few things. Insulin. What is insulin? We know that insulin is a polypeptide hormone which is synthesized by the isolate of Langerhans of the pancreas. Which cell? Of course, the beta cell. Now, the main metabolic action of this hormone is to controlling the blood glucose level. Now, this for this particular purpose, the insulin having the activity into the metabolism of the carbohydrate, protein and fat. For example, it is including the glycolysis, uh, including inclusion of the glucose into the glycogen molecule, transforming the glucose in lipid and protein. Of course, the glucose uptake into the fat tissue and all, skeletal muscle, etc., etc. Now we know that the different types of the hormones are secreting from the isolate of Langerhans, like glucagon, which is increasing the blood sugar, somatostatin and the insulin. But insulin we are mainly considered as this is the main hormones which is controlling the blood glucose level in our body. Now what is mean by the diabetes mellitus? Diabetes mellitus is a metabolic disorder which is occurred due to the improper metabolism of carbohydrate, protein or the fat and characterized by the hyperglycemia. What is hyperglycemia? Increase into the blood glucose level. Now there is a mainly two type of the diabetes mellitus. Type 1, insulin dependent diabetes mellitus. What will happen? In this case, insulin beta cell it will be destroyed. So there is a insufficient synthesis of the insulin. And in case of the type 2, it may be occur into the two category, either the reduced insulin secretion or the insulin resistance. That means insulin it will be there, the body cells it will not responding towards the insulin, but both the cases the blood glucose level it will be increases. And some other diabetes mellitus category also is there, like gestational diabetes mellitus which is occur during the pregnancy and the other special. Now, what is the objective of this particular experiment is demonstration of effect of insulin on the healthy rats and on the rats on the insulin dependent diabetes mellitus produced by administering alaxone, a substance which destroys the cell from the isolate of Langerhans of the pancreas. Now, generally, whatever alaxone is a chemical which is destroying the beta cell. Now if the beta cell is destroyed, what will happen? The blood glucose level it will be increased, and as it is destroying the beta cell, it will produce the type 1 diabetes mellitus into the experimental animal. Now next we will see about the principle. Okay, principle. Okay, this is the procedure to collect the blood sample from the normal rats and then another blood sample from the rats with the diabetes mellitus before and after insulin administration, measure the blood glucose level. Now we need to remember that, see here the experiment is very easy. We need to click, we need to inject the alaxone, it will produce the diabetes mellitus, but actually it is not that much easy. What will happen? We are keeping the animal for a particular time, then we are injecting a specified amount of the alaxone, like you can say that 60 mg per kg or 150 mg per kg. And what will happen after injecting into the diabetes mellitus? We have to keep the animal under the supervision, because sometimes death also it will be there. after 48 hours or 72 hours, depend upon the guidelines, the animals, it will develop the type 1 diabetes mellitus. And if the blood glucose level rise more than 200, then we are considering them under this particular category, type 1 diabetes mellitus. And then we need to give the drug for a particular duration. For example, if you are giving the 1 or 2 dose, may not you get the good result. More 7 days, 14 days, 21 days, 28 days, 3 month study. So it may be the different study it is involved. So here actually, as it is a software, so we can see that what is the effect of this, you can say, the drug into uh, blood glucose level of the rat. So let's see how we can perform the experiment.